My name is Sabrina McNew, and I'll be talking about work that I'm doing for my PhD at the University of Utah. When faced with attack by parasites and pathogens, animals deploy a number of different defenses. These defenses can be categorized into two major categories of resistance and tolerance. Resistance mechanisms help the host by reducing parasite burden, and thus the host can maintain host fitness. So in this example, when attacked by these purple parasites, the bird could mount an immune response, and this would kill some of the parasites, and the host survives. In contrast, tolerance mechanisms are ways by which the host can recover for energy lost to the parasite without reducing parasite burden. In this example, the host could also increase cellular repair, which would allow it to maintain fitness without affecting the parasite burden. Now, resistance mechanisms have been well studied, but we're increasingly finding that tolerance is a common and effective defense. However, there's still a lot to learn in terms of what the particular mechanism of defense might be in any system, and also what resources are required to mount an effective defense. I'm studying tolerance in the Galapagos mockingbird, which is an endemic bird found in the Galapagos Islands off of the coast of mainland Ecuador. We work on Santa Cruz Island in the middle there, at a site along the coast indicated by the red star. The parasite in question is Florence downsii, which is an introduced parasitic nest fly in the Galapagos. The adult flies are not parasitic, but they lay their eggs in the nests of birds, and the three larval instars that you see on the right side, or left side, feed on nestling birds and their mothers before pupating in the nest, and then emerging as flies again, adult flies. P. downsii is a cause for concern in the Galapagos because it causes extremely high mortality in Darwin's finch nests. Here we can see examples of the pathology of parasitism. On the left-hand side, you can see a healthy chick. It's relatively plump and rosy, and you can contrast that with the heavily parasitized chick on the right side. Notice that it is smaller, it's anemic looking, and you can even see the lesions from the parasitism on the abdomen and in the nares of the bird. People started to notice Florence downsii in the Galapagos about 20 years ago when they saw chicks that looked like that sick chick on the right side. However, not all endemic species of birds in the Galapagos seem equally vulnerable to this fly. In fact, Galapagos mockingbirds show tolerance to Florence downsii. In 2 years of study led by my former lab mate, Sarah Canuti, we found that P. downsii did not affect mockingbird fledging success. And we think that mockingbirds were tolerant to this parasite because even though nestlings were heavily infested with the fly, they were able to survive. And we think the mechanism of tolerance was that parents of parasitized nests fed their nestlings more than parents of non-parasitized nests. So potentially increased provisioning allowed parasitized nestlings to recover from the energy lost to the parasite. But these data don't tell us whether nestlings are tolerant in all conditions. And why would we think that tolerance would vary? Well, if the mechanism of tolerance is increased provisioning, it might follow that tolerance depends on food availability and thus environmental conditions. The environment in the Galapagos is actually quite variable. Mockingbirds breed during the rainy season when larval abundance is highest, but both the timing and the duration of rains varies quite a bit from year to year. We can see differences qualitatively by comparing these two pictures that were taken at the same point in our field site. At the top you see the rainy conditions and in the bottom the dry conditions. We can also see differences quantitatively by looking at rainfall and EVI, which stands for Enhanced Vegetation Index, and is a measure of greenness based on satellite imaging. And in what we call dry years, both rainfall and EVI are about half of what's typical. So the first 2 years in which we saw that mockingbirds were tolerant to P. downsii both happened to be typical rainy years. But the last 2 years have been what we call dry years. And so we've been able to ask the question, is tolerance condition dependent? So how do we do this? We go out in our field site and we look for active mockingbird nests. Here is my lab mate Graham on a ladder looking into the nest of one of the mockingbirds. They nest in these tall tree cacti and other trees. And when the nestlings hatch, we briefly remove them while we spray the nest either with 1% permethrin, which is a dilute insecticide that kills Florence larvae, or with water, which serves as a control and allows the nest to be naturally infested. And prevalence in our system is about 100%, so basically all of our water-treated nests end up being infested. We place video cameras at a subset of nests so that we can record the behavior of nestlings and their parents. Here you can see the mockingbird parents diligently feeding a brood of hungry chicks. Then we return 2 more times to measure growth and condition of the nestlings. At the 3rd visit, we put color bands on the nestlings so that when we fledge, we can use resighting of color-banded fledglings. This one does have color bands underneath those feathers. We use fledging success as an indicator of fitness. So after the nestlings fledge or die, we take down the nest and we dissect the nest to look for Florence pupae and larvae. Remember, as I said, Florence pupae in the nest, and this means that we can gain an accurate assessment of the intensity of parasitism by dissecting the nest even after the fledglings leave. Moving on to some results. First, let's look at P. downsi abundance. Here on the y-axis, we're looking at the average number of parasites per nest over 4 years of study, 2 rainy years and 2 dry years. The N in each case is the total number of nests that we studied that year. The blue bars are those nests that were treated with water, that is the control. And the black bars that you can barely see at the bottom of the figure are those nests treated with permethrin. And I'll continue with this color scheme in the subsequent slides. So I want to point out 2 things here. The first thing is that our treatment is very effective. Those nests that are treated with permethrin have almost no parasites. And the second thing is that parasite abundance does not seem to vary much among years. That is, there's no significant difference between the average number of parasites per nest in rainy years versus dry years. Next, looking at the effect of parasitism on nestling condition, we find in every year parasitized nestlings are in worse condition. For example, here we can look at hemoglobin concentration, which is an indicator of anemia. Lower hemoglobin is lower condition. And here we're just comparing 3 years of data, 1 rainy year and 2 dry years. The letters over the bars indicate statistically significant differences. And we see here in every year, parasitized nestlings are in worse condition. This kind of makes sense. These nestlings are being parasitized by a lot of these little larval flies, and so they're losing blood to the parasitism, and that's reflected in the hemoglobin concentration. However, the ability of nestlings to recover from this blood loss does seem to vary among years. First, let's look at nestling mass, which is on the y-axis here, this time over the 4 years of our study. And we can see that in rainy years, there is no significant difference between treatments in nestling mass. So even though they're losing the blood to the parasite, they're still growing at comparable rates to the non-parasitized nestlings. However, in dry years, we see a dramatic difference in mass between treatments, and the parasitized nestlings are much smaller than non-parasitized nestlings, while non-parasitized nestlings are approximately the same size as nestlings from previous years. And ultimately, we see that the fitness effects of P. downsi and Galapagos mockingbirds are very different in rainy and dry years. Again, here we're comparing data from 4 years of study, and on the y-axis we see the total % fledging success per treatment. So in the rainy years, we see that there's no significant difference between treatments. That is, mockingbirds are tolerant, and most nestlings, whether or not they're parasitized, tend to survive. Whereas mockingbirds lose their tolerance in dry years, and we see very low survival of parasitized nestlings. In order to see if this loss of tolerance is related to behavior, we turned back to our videos and we scored a number of different nestling and adult behaviors. But I'll highlight provisioning behavior because we think that's particularly important. Here on the y-axis, I'm showing the average time that parents spent provisioning their nestlings over 2 years, a rainy year and a dry year. Like I said before, in the rainy year, when mockingbirds were tolerant, parasitized nestlings were fed more, and this is why we think they were able to survive. However, we saw in the dry year that there was no significant difference in provisioning rates between treatments. So potentially because the food resources were limited in the dry year, we saw a really high fitness effect of parasitism and this loss of tolerance. So I'd like to discuss these data and a number of different implications that we've seen. First of all, we found that Galapagos mockingbirds have a defense against a recently introduced parasite. And this defense seems to be increased provisioning, which allows them to compensate for energy lost to the parasite without decreasing parasite burden. However, we've seen that mockingbirds are not tolerant in all years. In years of worse environmental conditions, where supposedly food abundance is limited, they lose tolerance. This highlights the fact that host-parasite interactions are labile and depend on potentially other factors besides just the two species studied. This means that if we just look at a snapshot of a host-parasite interaction, we may miss some of the dynamics of what's going on in that system. This also means that in looking at future impacts of P. downsi on mockingbird populations, this may depend a lot on what the environment looks like in the future. And this is just yet another example of why, in studying host-parasite interactions and for conservation, it's important to learn more about the dynamics of climate variability and understand some of the potential future effects of global climate change. And lastly, we're beginning to talk about potential interactions between the two species that I talked about today and other hosts of this generalist parasite in the community, such as Darwin's finches. Remember, because tolerant hosts do not decrease the abundance of parasite, this means they are not selecting on the parasite for reduced fitness, and they can also maintain the population of the parasite in the community. This means that Galapagos mockingbirds, in effect, may be serving as a reservoir of infection, which could increase the intensity of infection on more vulnerable hosts, such as species of Darwin's finches. So in the future, we're hoping to be able to understand a little bit more about how tolerance in mockingbirds, or lack thereof, influences the effect of this invasion on other species that are important in the system. So there, I will end. I'd like to acknowledge the work done by our field team and thank the members of my lab. And I will take any questions that you guys have.
Did you notice anything happening with the individuals that pledged and joined? Was there something that just reduced parasites in these nests? There's variation of those nest clusters. Was there actually some kind of negative indication? Are the birds responding in kind of interesting way that might allow them to compensate for years of joins?
I think one thing that might be important is that there is some heterogeneity in our environment, and so potentially those nestlings that survived, although parasitized, were potentially in territories that were slightly better, or they had parents that were slightly better at finding food, and so that could be part of it. Thank you. Oh, one more question?
Any evidence of resistance mechanisms, either among nests or different species, or?
No, we've not found any evidence of resistance defenses in this system. Previous students in our lab have looked for an immune response to the parasite. The nestlings don't seem to be able to mount an immune response to the parasite. Probably they're too young. We did find that female medium ground finches do have an immune response to the parasite, and females get parasitized while they're incubating the nestlings and the eggs. And this was correlated with a decreased number of parasites in the nest, but it did not translate into increased fitness of the nestlings. Okay, thanks.